My respected brothers and sisters, my name is Dr. Fahmy, and I'm delighted to welcome you to today's session on pharmaceutical sterility as part of our pharmaceutical course. Throughout this session, we'll delve into the intricacies of ensuring sterility in pharmaceutical products, a critical aspect of maintaining their safety and efficacy. I hope that by the end of our discussion, you'll gain valuable insights into the importance of sterility in pharmaceutical manufacturing processes. So, without further ado, let's dive into our exploration of pharmaceutical sterility. Thank you for joining me, and let's get the ball rolling. Aseptic means contamination-free. It ensures that a product won't harbor harmful microorganisms like bacteria or viruses. Essentially, it prevents their growth and reproduction, crucial for maintaining product integrity during handling and storage. On the other hand, sterility takes it a step further. It means a product is entirely free of all germs. Achieving this status involves a process called sterilization, where all viable microorganisms, including bacteria, viruses, fungi, and others, are eliminated. Now, let's clarify the differences. Aseptic ensures a contamination-free environment and prevents microorganism reproduction. Sterile, however, means the product is completely devoid of all germs, achieved through sterilization. Understanding these distinctions is vital in maintaining product integrity and ensuring patient safety. So how do we achieve sterilization? Sterilization involves various methods such as heat, radiation, chemical sterilants, and filtration. Each method aims to eliminate all viable microorganisms present on surfaces or within substances. Let's explore the critical variables that impact the attainment of sterility in pharmaceutical products. Firstly, temperature and humidity levels during the sterilization process are paramount. Optimal conditions must be maintained to ensure effective sterilization. Deviations from these conditions can compromise the process and result in inadequate sterilization. Secondly, proper preparation and loading of the item into the sterilizer are essential. This ensures uniform exposure to the sterilant and minimizes the risk of contamination. Attention to detail in this step is crucial to achieve thorough sterilization outcomes. Thirdly, effective delivery of the sterilant into the system is vital. Proper penetration and contact with all surfaces of the item being sterilized are necessary for successful sterilization. Any shortcomings in sterilant delivery can lead to incomplete sterilization and pose risks to product safety. Fourthly, adherence to the maintenance protocol is imperative. Regular maintenance of sterilization equipment ensures consistent performance and prevents malfunctions that could compromise sterility. Following the prescribed maintenance procedures diligently is essential for maintaining the integrity of the sterilization process. Lastly, utilizing the correct cycle and sterilization procedure is crucial. Different items may require specific parameters to achieve optimal sterilization results. It's essential to select the appropriate cycle and procedure tailored to the item being sterilized to ensure effectiveness and safety. Let's delve into the various types of sterilization methods used in pharmaceutical science to ensure the safety and efficacy of medical products. Firstly, we have high temperature and pressure sterilization, often carried out using a steam autoclave. This method involves subjecting the items to high temperatures, typically 121 degrees Celsius, and pressure, under 15 psi. The combination of heat and pressure effectively kills microorganisms, ensuring thorough sterilization of the materials. Secondly, chemical sterilization offers an alternative approach. Various chemicals such as ethylene oxide, chlorine dioxide, and ozone are utilized in this method. These chemicals work by disrupting the vital functions of microorganisms, ultimately leading to their destruction. Chemical sterilization is particularly useful for heat-sensitive materials that cannot withstand high temperatures. Next, we have radiation sterilization, which utilizes different forms of radiation, such as gamma rays, X-rays, and electron beam. These high-energy radiations penetrate the materials, damaging the DNA and other vital components of microorganisms, thereby rendering them sterile. Radiation sterilization is widely used for items that cannot be sterilized using heat or chemicals. Lastly, filtration sterilization is another commonly employed method. This method involves passing the solution or suspension through a filter with pores small enough to trap and remove microorganisms. Filtration is particularly effective for sterilizing liquids or gases and is often used in pharmaceutical manufacturing processes. By employing these diverse sterilization techniques, pharmaceutical scientists can ensure the safety and efficacy of medical products, ultimately contributing to the improvement of human health worldwide. Now let's explore the steam autoclave, an essential tool in the realm of sterilization, particularly in medical settings. Firstly, the steam autoclave stands as one of the earliest, safest, and most cost-effective methods of sterilization, especially in medical applications. Its reliability and efficiency have made it a cornerstone in ensuring the safety of medical equipment and instruments. Operating at 121 degrees Celsius and under 15 pounds per square inch, (psi), of pressure for a duration of 15 to 60 minutes, the steam autoclave employs a combination of high temperature, pressure, and steam to eliminate microorganisms effectively. The process is straightforward yet highly effective. The intense heat, pressure, and hot steam work in tandem to eradicate microorganisms, creating conditions of sterility within the autoclave chamber. In medical applications, steam autoclaves are widely utilized to sterilize surgical instruments, ensuring that they are free from any potential pathogens before use in surgical procedures. The reliability and efficacy of this method have made it a standard practice in healthcare facilities worldwide. However, it's essential to note that steam autoclaving may not be suitable for items that are heat and moisture sensitive. The high temperatures and steam used in the process may cause damage to such materials, rendering them unsuitable for autoclave sterilization. In general, the steam autoclave remains a cornerstone in medical sterilization, offering a safe, reliable, and cost-effective means of ensuring the sterility of medical instruments. While highly effective for most applications, it's important to consider the suitability of materials before subjecting them to autoclave sterilization. Let's delve into the fascinating concept of the Z-value in thermal processing, a crucial aspect of sterilization and preservation in various industries, including pharmaceuticals and food. The Z-value of an organism represents the temperature, in degrees Fahrenheit, required for the thermal destruction curve to move one log cycle. While the D-value gives us the time needed at a certain temperature to kill an organism, the Z-value provides insight into the organism's resistance to temperature variations. In essence, the Z-value enables us to calculate thermal process equivalency. For instance, if it takes a 10-degree Fahrenheit increase to move the curve one log, then our Z-value is 10. Armed with this information, we can calculate D-values for different temperatures by adjusting the time accordingly. Let's consider an example. If we have a D-value of 4.5 minutes at 150 degrees Fahrenheit, we can calculate the D-value for 160 degrees Fahrenheit by reducing the time by one log. Thus, our new D-value for 160 degrees Fahrenheit would be 0.45 minutes. This illustrates that each 10-degree Fahrenheit increase in temperature reduces the D-value by one log. Conversely, a 10-degree Fahrenheit decrease in temperature would increase the D-value by one log. Therefore, if we were to consider a temperature of 140 degrees Fahrenheit, the corresponding D-value would be 45 minutes. In conclusion, understanding the Z-value allows us to comprehend the thermal resistance of microorganisms and calculate the necessary adjustments in time and temperature for effective sterilization and preservation processes. We are going to explore alternative methods of sterilization, particularly focusing on ethylene oxide gas and vaporized hydrogen peroxide, VHP, which offer effective solutions for sterilizing heat and moisture-sensitive items. Ethylene oxide gas sterilization involves the use of a gas with low temperature, typically ranging from 50 to 60 degrees Celsius. Unlike traditional steam autoclaves, this method operates at lower temperatures, making it suitable for delicate and sensitive materials that cannot withstand high heat. However, it's important to note that ethylene oxide gas sterilization may require a longer time to reach the desired sterility condition due to its lower temperature. Despite this drawback, it remains a valuable option for ensuring the safety of heat and moisture-sensitive items in various industries, including healthcare and pharmaceuticals. On the other hand, vaporized hydrogen peroxide, VHP, sterilization offers another alternative for sterilizing sensitive items. In this method, hydrogen peroxide is vaporized and injected into the sterilization chamber, where it acts as a powerful sterilant. The cycle time for vaporized hydrogen. Peroxide sterilization typically lasts around 2 hours, with temperatures ranging from 30 to 40 degrees Celsius. While this method also operates at lower temperatures, it provides effective sterilization without compromising the integrity of sensitive materials. Both ethylene oxide gas and vaporized hydrogen peroxide, VHP, sterilization offer viable alternatives to traditional steam autoclaves, particularly for heat and moisture-sensitive items. Despite requiring longer cycle times and operating at lower temperatures, these methods provide reliable sterilization solutions, ensuring the safety and integrity of sensitive materials in various industries. Now let's explore the fascinating world of radiation sterilization, a highly effective method utilized in various industries, including healthcare and food processing. Radiation sterilization encompasses several forms of radiation, including gamma rays, X-rays, and electron beam radiation. Each of these methods offers unique advantages and applications in sterilizing a wide range of materials and products. X-rays, for instance, provide excellent product penetration during sterilization. This means they can effectively treat both the surface and interior parts of items, ensuring thorough sterilization throughout. Gamma radiation, on the other hand, typically requires exposure, times ranging from 10 to 20 hours, depending on the strength of the exposure. Despite the longer exposure times, gamma radiation remains a reliable method for achieving sterilization, particularly for items that are heat or moisture sensitive. During radiation sterilization, temperatures typically reach a range of 30 to 40 degrees Celsius. While not as high as those used in steam autoclaves, these temperatures are sufficient to ensure the effectiveness of the sterilization process. The mechanism behind radiation sterilization lies in its ability to disrupt the DNA materials of microorganisms. By damaging the DNA, radiation effectively kills microorganisms, thereby achieving sterility in the treated items. So radiation sterilization, whether through gamma rays, X-rays, or electron beam radiation, offers a powerful means of achieving sterility in various materials and products. With its ability to penetrate surfaces and disrupt microorganisms' DNA, radiation sterilization stands as a cornerstone in ensuring the safety and integrity of medical devices, pharmaceuticals, and other sensitive products. Now let's explore the process of filtration sterilization, a vital technique employed in various industries, particularly in pharmaceuticals and biotechnology. Filtration sterilization involves passing liquids or gases through filters with pores smaller than 0.22 micrometers. These filters act as barriers, trapping and removing microorganisms, ensuring that the final product is free from contamination. Primarily used for liquids utilized in cell cultures and microbial studies, filtration sterilization offers a reliable method for maintaining the purity of these critical materials. By removing bacteria, viruses, and other contaminants, it helps to safeguard the integrity of experiments and production processes. Moreover, filtration sterilization extends its utility to gases, which may be used to purge sterile chambers or release vacuum in lyophilization chambers. By filtering out any potential contaminants, it ensures the sterility of the environment, crucial for maintaining product quality and safety. However, the effectiveness of filtration sterilization relies heavily on the integrity of the filters themselves. Before initiating the filtration process, it is essential to verify the integrity of the filters to ensure they can withstand the pressure and effectively trap microorganisms. Additionally, it's crucial to reconfirm the integrity of the filters after the filtration process to guarantee their continued effectiveness. Hence, Filtration sterilization serves as a cornerstone in ensuring the purity and sterility of liquids and gases used in various applications, from pharmaceutical manufacturing to biotechnological research. By employing precise filtration techniques and verifying filter integrity, we can maintain the highest standards of product quality and safety. Let's talk about the different categories of sterile products, each with its unique approach to achieving sterility, ensuring the safety and efficacy of medical and pharmaceutical products. Firstly, we have sterile products where sterilization occurs directly within the final container. This method, known as terminal sterilization, involves subjecting the product and its container to sterilization processes simultaneously. This ensures that the product remains sterile until it is ready for use, providing a high level of assurance regarding its safety and effectiveness. However, there are certain products where terminal sterilization is not feasible or practical. In such cases, the products must be aseptically prepared. This means that while the product itself cannot undergo terminal sterilization, strict aseptic techniques are employed throughout the manufacturing process to prevent contamination. Despite the challenges, aseptic preparation allows for the production of sterile products without compromising their quality. To ensure the sterility of products prepared aseptically, stringent measures are implemented, including the adherence to Sterility Assurance Level, SAL. SAL represents the probability of a single unit of the product being non-sterile. Achieving the desired SAL requires the implementation of aseptic operations and practices, including the use of sterile equipment, environments, and processes. By diligently practicing aseptic techniques and adhering to SAL requirements, manufacturers can effectively achieve sterility assurance for products prepared aseptically, ensuring their safety and efficacy for use in healthcare settings. In conclusion, the categories of sterile products encompass both those that undergo terminal sterilization in their final container and those that require aseptic preparation. Each approach presents its challenges and requirements, but both are essential for ensuring the safety and effectiveness of sterile products in medical and pharmaceutical applications. Now, regarding aseptic processing, a revolutionary technique in the realm of food and pharmaceutical industries. Aseptic processing involves the packaging of commercially thermally sterilized liquid products, such as food or pharmaceuticals, into previously sterilized containers under sterile conditions. This innovative method yields shelf-stable products that do not require refrigeration, offering convenience and versatility in storage and distribution. Over time, aseptic processing has emerged as the preferred method, gradually replacing traditional container sterilization techniques for liquid foods. This transition encompasses a wide range of products, including milk, fruit juices and concentrates, cream, yogurt, salad dressing, liquid egg, and even ice cream mix. Moreover, the popularity of aseptic processing extends beyond traditional liquid foods to include items containing small discrete particles. From cottage cheese to baby foods, from tomato products to fruit and vegetables, from soups to rice desserts, aseptic processing offers a viable solution for preserving these diverse food items while maintaining their quality and freshness. The beauty of aseptic processing lies in its ability to sterilize the food outside the container, ensuring thorough sterilization, and then transferring it into a previously sterilized container. This entire process occurs in a sterile environment, guaranteeing the integrity and safety of the final product. Hence, aseptic processing represents a remarkable advancement in food preservation and packaging technology. Its ability to produce shelf-stable products without the need for refrigeration has revolutionized the food industry, offering consumers a wider variety of convenient and safe food options. Now, the concept of terminal sterilization, a crucial process in pharmaceutical manufacturing that ensures the safety and efficacy of sterile drug products. Terminal sterilization involves the sterilization of products within their final container. In the realm of pharmaceutical manufacturing, this process is commonly used for traditional liquid sterile drug products. These products are filled into clean and sterile primary containers, such as vials, bottles, syringes, cartridges, or bags. Following the filling process, the containers are sealed using primary and secondary packaging components. These may include pre-sterilized elastomeric stoppers, aluminum seals or closures, screw caps, or heat crimping methods. These components play a vital role in maintaining the sterility of the product until it reaches the end user. While stringent controls, including current good manufacturing practices, CGMP, are implemented throughout the manufacturing process to ensure the finished product's sterility, it's important to note that no sterility assurance level, SAL, is obtained from this process. Instead, the sterility of the drug product released from this type of manufacturing process relies entirely on the effectiveness of the manufacturing controls in place at the time of filling. In essence, terminal sterilization represents a critical step in pharmaceutical manufacturing, safeguarding the integrity and safety of sterile drug products. Through meticulous adherence to CGMP and rigorous manufacturing controls, pharmaceutical companies ensure that each product meets the highest standards of quality and sterility, providing patients with safe and effective medications. Let's talk about the critical process of validating sterility and aseptic conditions in pharmaceutical manufacturing, ensuring the safety and efficacy of medical products. Validation of sterility and aseptic conditions is paramount, designed to provide assurance regarding the integrity and safety of pharmaceutical products. This validation process involves specific and appropriate testing methodologies aimed at verifying that sterility and aseptic conditions are maintained and controlled within predetermined parameters. Throughout the validation process, various components are evaluated, including drugs, containers, as well as the processes and procedures involved in manufacturing. Each element undergoes rigorous scrutiny to ensure compliance with established standards and regulations, safeguarding the quality and safety of the final product. Specific pharmaceutical products require stringent validation procedures due to their critical nature. These include injections, ophthalmic preparations, irrigation solutions, and hemodialysis solutions, among others. Given their direct administration into the body, maintaining sterility and aseptic conditions is essential to prevent the risk of contamination and adverse effects on patients. Validation of sterility and aseptic conditions involves a combination of tests, including microbial testing, environmental monitoring, and process simulations. These tests ensure that the manufacturing environment remains free from microbial contamination and that the sterilization processes are effective in eliminating microorganisms. Ultimately, the validation of sterility and aseptic conditions is a comprehensive process aimed at guaranteeing the safety and efficacy of pharmaceutical products. By adhering to strict validation protocols and conducting thorough testing, pharmaceutical manufacturers can uphold the highest standards of quality and ensure the well-being of patients worldwide. Now, one of the important elements of sterility is the fundamental requirements in process validation, which is a crucial aspect of ensuring the quality and consistency of pharmaceutical manufacturing processes. Firstly, written documentation serves as the cornerstone of process validation. Detailed documentation outlines the entire manufacturing process, including procedures, protocols, and specifications. This documentation provides a roadmap for ensuring that each step is executed correctly and consistently, maintaining product quality and safety. Secondly, manufacturing parameters play a pivotal role in process validation. These parameters include variables such as temperature, pressure, time, and equipment settings. It's essential to establish and monitor these parameters to ensure that the manufacturing process operates within predefined limits, guaranteeing the reproducibility and reliability of the final product. Thirdly, testing parameters are integral to process validation. These parameters encompass the criteria used to evaluate the quality and performance of the manufacturing process. By defining specific testing methodologies and acceptance criteria, manufacturers can assess whether the process consistently meets regulatory requirements and product specifications. Moreover, an in-process protocol is essential for process validation. This protocol outlines the steps and procedures for monitoring and controlling the manufacturing process in real time. By implementing an in-process protocol, manufacturers can identify and address any deviations or issues promptly, minimizing the risk of producing non-compliant or substandard products. Lastly, final product testing serves as the ultimate validation of the manufacturing process. Through rigorous testing and analysis, manufacturers verify that the final product meets all required specifications and quality standards. This ensures that the product is safe, effective, and fit for its intended use. So, process validation is essential for ensuring the quality, safety, and consistency of pharmaceutical products. By adhering to basic requirements such as written documentation, monitoring manufacturing parameters, defining testing parameters, implementing in-process protocols, and conducting final product testing, manufacturers can uphold the highest standards of quality assurance and regulatory compliance. Let's delve into the critical process of validating steam autoclaves, an essential step in ensuring the effectiveness and reliability of sterilization in various industries, including pharmaceuticals and healthcare. The validation of steam autoclaves involves several key considerations, each aimed at confirming the sterilization process's efficacy and consistency. Firstly, heat distribution within the sterilization chamber is of utmost importance. Thermocouples, specialized temperature sensors, are strategically distributed and evenly placed throughout the chamber to monitor temperature distribution. This ensures that heat is evenly distributed, reaching all areas of the chamber and guaranteeing thorough sterilization. Secondly, maintaining precise temperature control is essential. The temperature deviation should not exceed plus 2.5 degrees Celsius of the mean chamber temperature. This tight control ensures that the sterilization process operates within specified parameters, minimizing the risk of inadequate sterilization or overheating. Furthermore, heat penetration studies are conducted to assess the steam autoclave's ability to penetrate and effectively sterilize materials. Bacillus stearothermophilus and Clostridium sporogenes are commonly used as biological indicators to challenge moist heat sterilization. These studies provide critical insights into the autoclave's performance and help verify its effectiveness in sterilizing various materials. In recent advancements, thermal tape has emerged as a convenient and reliable indicator of sterilization. This specialized tape changes color when exposed to the appropriate temperature and pressure conditions, providing a visual confirmation of successful sterilization. Thermal tape serves as an additional layer of assurance, complementing the data collected from thermocouples and biological indicators. Hence, the validation of steam autoclaves is essential for ensuring the safety and efficacy of sterilization processes in pharmaceutical and healthcare settings. By adhering to rigorous validation protocols, including monitoring heat distribution, controlling temperature deviations, conducting heat penetration studies, and utilizing reliable indicators such as thermal tape, manufacturers can maintain the highest standards of sterility assurance and regulatory compliance. Now let's explore the vital process of validating gaseous sterilization, a crucial step in ensuring the safety and efficacy of sterilization processes in various industries, including healthcare and pharmaceuticals. Validating gaseous sterilization involves several key considerations, each aimed at confirming the effectiveness and reliability of the sterilization process. Firstly, it's essential to consider the product specification and packaging design. Different products and packaging materials may require specific sterilization parameters to ensure thorough sterilization without compromising product integrity. By carefully analyzing product specifications and packaging designs, manufacturers can tailor the sterilization process to meet the unique needs of each product. Secondly, calibration verification of equipment is critical. Accurate calibration of sterilization equipment ensures that it operates within specified parameters, delivering consistent and reliable sterilization results. Regular calibration verification helps maintain the accuracy and reliability of equipment, minimizing the risk of under- or over-sterilization. Furthermore, verification on biological samples and thermocouples monitoring is essential. Biological indicators, such as spore strips or ampules containing spores, are used to challenge the sterilization process and verify its effectiveness in eliminating microbial contaminants. Thermocouples, specialized temperature sensors, are strategically placed to monitor temperature distribution throughout the sterilization chamber, ensuring uniform heating and sterilization. In addition to biological indicators and thermocouples, trial runs are conducted to validate the sterilization process. These trial runs involve running the sterilization cycle with dummy loads to simulate real-world sterilization conditions. By analyzing the results of trial runs, manufacturers can assess the effectiveness of the sterilization process and make any necessary adjustments to optimize performance. In general, validating gaseous sterilization is essential for ensuring the safety and efficacy of sterilization processes in healthcare and pharmaceutical settings. By considering product specifications and packaging designs, calibrating equipment, verifying biological samples and thermocouples monitoring, and conducting trial runs, manufacturers can maintain the highest standards of sterility assurance and regulatory compliance. The critical process of validating radiation sterilization, which is a fundamental step in ensuring the safety and efficacy of sterilization processes, particularly in industries such as healthcare and pharmaceuticals. Validating radiation sterilization involves several key considerations, each aimed at confirming the effectiveness and reliability of the sterilization process. Firstly, it's essential to consider the product specification and packaging dimensions. Different products and packaging materials may require specific radiation doses and exposure times to ensure thorough sterilization without compromising product integrity. By carefully analyzing product specifications and packaging dimensions, manufacturers can tailor the sterilization process to meet the unique needs of each product. Secondly, agreement and licensing of the equipment and radiation sources are crucial. Radiation sterilization facilities must adhere to strict regulations and obtain appropriate licenses to operate. This ensures that the equipment and radiation sources meet safety standards and are capable of delivering the required sterilization doses reliably and consistently. Furthermore, calibration of the dosimeter system is essential. Dosimeters are used to measure the radiation dose received by the product during sterilization. Calibration of the dosimeter system ensures its accuracy and reliability, providing confidence in the sterilization process's effectiveness. Moreover, attention must be paid to dose, rate, and strength parameters. The radiation dose, rate of exposure, and strength of the radiation source are critical factors that influence the effectiveness of sterilization. Manufacturers must carefully control these parameters to ensure that the required sterilization dose is delivered uniformly and consistently throughout the product. All in all, validating radiation sterilization is essential for ensuring the safety and efficacy of sterilization processes in healthcare and pharmaceutical settings. By considering product specifications and packaging dimensions, ensuring agreement and licensing of equipment and radiation sources, calibrating the dosimeter system, and controlling dose, rate, and strength parameters, manufacturers can maintain the highest standards of sterility assurance and regulatory compliance. Regarding the critical process of validating filter sterilization, this process involves several key considerations, each aimed at confirming the filter's ability to remove bacteria and maintain sterility. Firstly, validation of the filter is paramount. Manufacturers must demonstrate the filter's ability to effectively remove bacteria and other microorganisms from the fluid being filtered. This validation process ensures that the filter meets the required standards for microbial retention and sterility. Secondly, the filter's ability to retain specific bacteria is assessed. Brevundimonas diminuta, a commonly used test organism, is often employed to challenge the filter's performance. The filter must demonstrate its ability to retain Brevundimonas diminuta with a high bacterial load of 10^7 colony-forming units, CFU, per square centimeter of the filter surface. This rigorous test ensures that the filter can effectively capture and retain bacteria, maintaining the sterility of the filtered product. Furthermore, validation of filter sterilization requires real-world testing. The items that need to be filtered are mixed with the test bacteria to simulate actual filtration conditions. This ensures that the filtration process performs as expected under practical circumstances, validating its effectiveness in removing bacteria and maintaining sterility. So, validating filter sterilization is essential for ensuring the safety and efficacy of filtration processes in pharmaceutical and biotechnology applications. By validating the filter's ability to remove bacteria, retaining Brevundimonas diminuta under high bacterial loads, and conducting real-world testing with test bacteria, manufacturers can ensure that their filtration processes meet the highest standards of sterility assurance and regulatory compliance. Now we have to discuss about the various grades of clean areas within manufacturing environments, each serving a specific purpose in maintaining the cleanliness and sterility required for pharmaceutical and healthcare operations. Firstly, we have Grade D clean areas, also known as Class 100,000 or ISO 8 environments. These areas are suitable for manufacturing processes with less critical stages aseptically, such as handling components after washing. While not as stringent as higher grades, Grade D clean areas still play a crucial role in ensuring the cleanliness and integrity of the manufacturing process. Moving up the scale, we have Grade C clean areas, designated as Class 10,000 or ISO 7 environments. These areas are suitable for manufacturing processes, with slightly more critical stages aseptically, such as the preparation of solutions to be filtered. Grade C clean areas provide a higher level of cleanliness and sterility compared to Grade D, making them ideal for processes requiring greater precision and control. Next, we have Grade B clean areas, classified as Class 1000 or ISO 6 environments. These areas serve as the background environment for more critical working areas, such as clean rooms equipped with laminar flow systems. Grade B clean areas provide enhanced cleanliness and sterility, creating an optimal environment for sensitive manufacturing processes. Finally, we have Grade A clean areas, characterized as Class 100 or ISO 5 environments. These areas represent the highest level of cleanliness and sterility within manufacturing environments. Grade A clean areas serve as the background environment for the most critical working areas and high-risk operations, such as product filling, handling sterile materials, and aseptic processes. They are meticulously maintained to ensure the utmost integrity and safety of pharmaceutical and healthcare products. In conclusion, the various grades of clean areas within manufacturing environments play a vital role in maintaining the cleanliness, sterility, and integrity required for pharmaceutical and healthcare operations. By adhering to stringent cleanliness standards and utilizing appropriate clean area classifications, manufacturers can ensure the safety, efficacy, and quality of their products. Now move on to the importance of airflow patterns in environmental monitoring, particularly in critical areas within pharmaceutical and healthcare facilities, where maintaining cleanliness and sterility is paramount. First and foremost, airflow over critical areas should be unidirectional. This means that air should flow in a single direction, preventing the recirculation of contaminants and ensuring that any particles are carried away from sensitive areas, such as filling or closing zones. Furthermore, the velocity of airflow should be strong enough to effectively sweep particles away from these critical areas. A recommended velocity of approximately 0.45 meters per second, with a tolerance of plus 20%, is typically employed. This airflow velocity, coupled with a positive pressure of 10 to 15 pascals, helps to create an environment where contaminants are efficiently removed from the workspace. Maintaining positive pressure between adjacent rooms of different classifications is crucial. This prevents the migration of contaminants from lower classified areas to higher classified ones, preserving the integrity of sterile environments. In facilities with multiple rooms, it's essential to ensure that the most critical areas or rooms have the highest pressure. Continuous monitoring and observation of pressure differentials are necessary to detect any deviations from the desired levels. Any significant deviations in pressure will trigger an alarm, prompting immediate corrective action to maintain the integrity of the sterile environment. So, airflow patterns play a vital role in environmental monitoring within pharmaceutical and healthcare facilities. By ensuring unidirectional airflow, sufficient velocity, and appropriate positive pressure, Manufacturers can minimize the risk of contamination and maintain the sterility of critical areas. Continuous monitoring and prompt response to deviations in pressure are essential for safeguarding product quality and patient safety. Now, some essential tips for maintaining aseptic conditions in pharmaceutical and healthcare settings, where cleanliness and sterility are paramount to ensure the safety and efficacy of products and procedures. First and foremost, it's crucial to minimize the number of personnel in clean areas, especially during aseptic processing. Limiting the presence of individuals helps reduce the risk of introducing contaminants into the environment, preserving the sterility of critical areas. Continuous training of personnel and staff is essential. This includes comprehensive training on cleaning and maintenance procedures to ensure that all employees understand and adhere to stringent cleanliness protocols. Regular training sessions help reinforce best practices and promote a culture of cleanliness and sterility throughout the facility. Furthermore, decontamination procedures are necessary, particularly when personnel handle biological samples such as animals and bacteria. Implementing effective decontamination protocols helps prevent the spread of potentially harmful microorganisms, minimizing the risk of contamination in clean areas. Observing the usage of outdoor and indoor clothing is also crucial. Personnel should change their clothing regularly, at least daily or before each working session, to prevent the introduction of contaminants from external environments. Additionally, maintaining separate laundry facilities for cleanroom garments further reduces the risk of cross-contamination. Lastly, it's essential to change gloves and masks for every working session. These protective gear items serve as barriers against contaminants and should be replaced regularly to maintain their effectiveness. By changing gloves and masks frequently, personnel can minimize the risk of contaminating sterile environments and ensure the integrity of aseptic conditions. Last but not least, let's delve into the critical aspect of personnel monitoring in pharmaceutical and healthcare settings. Firstly, let's discuss the glove fingertips procedure, a fundamental aspect of personnel monitoring. This procedure involves regularly validating the presence of any microbial growth on the fingertips of gloves worn by personnel. By conducting this validation process, facilities can assess the effectiveness of hand hygiene practices and identify any potential sources of contamination. Regular monitoring of glove fingertips helps ensure that personnel maintain proper hygiene protocols and minimize the risk of introducing contaminants into sterile environments. Secondly, the gowning process plays a crucial role in personnel monitoring. Proper validation of the gowning process is essential to ensure that personnel wear appropriate protective garments effectively. By validating the gowning process, facilities can verify that personnel are adequately protected against contamination and adhere to established cleanliness protocols. This validation process helps maintain the integrity of sterile environments and reduces the risk of product contamination during manufacturing or handling procedures. Furthermore, it's essential to observe all procedures and workflows related to personnel activities. This includes monitoring personnel as they perform their tasks, ensuring that they follow established protocols and guidelines. By observing procedures and workflows, facilities can identify any deviations from standard practices and address them promptly. Continuous monitoring of personnel activities helps maintain the highest standards of cleanliness and sterility, safeguarding the integrity of pharmaceutical and healthcare products and procedures. In conclusion, personnel monitoring is a crucial component of maintaining cleanliness and sterility in pharmaceutical and healthcare settings. By implementing procedures such as glove fingertips, validating the gowning process, and observing all procedures and workflows, facilities can ensure that personnel adhere to strict cleanliness protocols and minimize the risk of contamination. Through continuous monitoring and adherence to established guidelines, facilities can uphold the highest standards of product quality and safety. Thank you all for your attention during this session. I trust that you found the information shared here valuable and insightful. Have a wonderful day ahead, and until next time, take care and cheers.